Hello and welcome. My name is Anthony Taylor and I'll be discussing with you today my ongoing research into the response of white Dutch clover to the organic molecule cadaverine and its potential forensic applications. First off, an introduction. Why undertake this research? Let's start with the background. Baltimore City, like most cities, experiences crime, and a subset of crime involves the unlawful death of an organism, often resulting in a body that is then hidden in what forensic investigators call a clandestine grave. A clandestine grave is defined as a grave site that is dug secretly and kept secret because the contents are the result of an illicit action. From the standpoint of law enforcement, handling the discovery of these sites is labor-intensive. When a suspected clandestine grave is brought to the police's attention, it is standard operating procedure for a crime scene investigator to be called in to process the site. This process requires a huge amount of documentation and may take up to several days to complete. As time is money, as well as an evidence-destroying force, quick and effective evidence-gathering methods are paramount to forensic investigators. There exists a subset of forensic science called forensic biology, which deals with obtaining forensic information from plants and animals involved with the scene. My interest is forensic botany, which is the forensic use of plants. It has many applications, and one is the detection and analysis of clandestine graves, because plants respond in a myriad of ways to the environmental disturbance caused by the digging and deposition of a novel nutrient source. However, as of 2020, police departments do not consider plant-based information about clandestine graves to be worth their evidentiary value and labor costs, and so no plant evidence is gathered or used in the state of Maryland. This also means that funding for research work involved with forensic botany is scarce at best, as few to no institutions are actually interested in the results. I believe this to be an oversight. There is definitely research potential here. Previous work has noticed interactions between plants and some of the large molecules associated with cadaveric decay. For example, legumes, such as chickpeas and soy, can absorb the large organic molecule cadaverine, a molecule strongly associated with decaying bodies, from the surrounding soil, and certain plant species will actually synthesize cadaverine in response to stressors like too much salt. In addition, forensic investigators for decades have noticed that the presence of a decomposing body tends to result in a circle of plant death surrounding the corpse. Despite these observations, to date, no one seriously looked into why these things occur. So I decided to investigate them myself. I began this research during the spring 2020 semester under Drs. Blome and Hastings, but unfortunately, due to COVID-19, the only data I managed to get was the final root and shoot mass of my first trial. However, it did look like there was a significant difference in the root size in cadaverine-treated plants versus the control, which gave me the proof of concept I needed to move forward with my research this summer. This topic is barely investigated and any data collected is novel, so what exactly have I been doing, and what have I found? Well, it's been all field studies in light of no lab access. I planned for 4 total experiments, one for growth, one for germination, a depth test, and a whole cadaver test, each with a focus question that I was trying to answer. Green text are experiments that have been completed at the time of this recording, and orange text means that it's still in progress. All 4 experiments deal with 2 main variables, a cadaverine source and a soil type. The various combos of these variables simulate a range of situations to gather data from. If you're interested in my experimental design and/or my reasoning behind things, please feel free to ask during the Q&A. I think we're all mostly interested in the results though, so let's move on. First up, the growth test. Pictured are my 36 treatment pots, day 1 of the 2-week trial on the left and the day of the harvest on the right. So I had a couple of significant results with this trial, but the biggest takeaway was that soil type had a much more substantial effect on plant growth than the cadaverine source. This actually surprised me because most forensic literature associates decay with plant death or stunting, so I was expecting that plants growing on a cadaverine source would be significantly smaller in mass than the control, but instead it seems its size was more closely linked to soil type. I also expected the roots to be more affected than the shoots, as increased cadaverine presence has been noted to trigger increased root growth, an observation that I also noticed in my work last semester. However, I do acknowledge I am not using pure cadaverine in these trials. There's lots of factors at play, and I also know that the effects of soil on plant health is often underestimated, so it may not actually be that surprising that these results do not actually track with what the literature has noticed. Here's a graph looking at soil's effects on plant growth. Plants in all other soils grew significantly better than the straight Maryland clay, but did not significantly differ from each other within this group. I was expecting this result from topsoil and topsoil humus, but the fact that the Maryland clay humus mix is so much better than the plain Maryland clay is interesting, as it may mean that plants growing in the natural Maryland clay soil that's been mixed with the more fertile top layer due to the digging of a grave will grow more robustly than plants in the surrounding area. As far as forensics is concerned, this may mean that clover growing on graves will be bigger and easier to spot as compared to nearby clover, which may assist in grave ID. From a botany perspective, this doesn't really add much new knowledge, as we're already aware that plants do poorly in pure red clay soil. These results are the same for both fresh and dry shoot weight, as shown by the above graph, which simply means that this effect is truly seen in the plant mass and is not skewed by water weight. Now let's discuss the germination test. Pictured again are my 36 treatment pots. On the left is day 1 of the 2-week trial, and on the right is the day of harvest. For this test, I also had a couple of significant results, which I'll describe in more detail in a second. All pots only took 3 days to germinate after seeding, but it became obvious very quickly that all treatment pots had a circular patch of 0 germination, whereas the control did not. Curious, I let them grow for a week and then tested the pH of the soil both within and outside the zone to look for differences, as I had a suspicion that maybe the decomposing organic matter was changing the soil pH. The pH inside the zone of inhibition was significantly affected by cadaverine source and to some extent soil type, seeming to trend towards an alkaline reading for both. Cheese was more alkaline than liver, although not significantly so, and Maryland clay was the most alkaline soil. This opens up the avenue that lack of germination on grave sites may be strongly tied to pH, specifically pH readings that are more alkaline than the average soil pH of 7. Outside the zone, it seems that any pH differences were due to soil type alone. Forensically, perhaps soil pH tests could help ID potential clandestine grave sites from just random dead patches that look sketchy. From a botanical standpoint, there's some real meat here for further studies on how pH affects clover germination and the germination of other leguminous plants. The size of the zone of inhibition was also extremely significant, and for this cadaverine source was the major player, with each treatment having a significantly different zone diameter. Liver was the biggest, then cheese, and then the control with none. From a forensic standpoint, this greatly pushes diameter of a dead patch as a visual cue that there's a clandestine grave. From a botanical standpoint, this is so cool because this suggests that there's definitely a tie between what's decaying and the extent of germination inhibition, something that there's been pretty much no work on. So this brings me to future directions. What does this mean? What can I do? My data established that soil seems to affect clover's growth more so than the presence of cadaverine. However, looking at the raw data, the source of cadaverine does seem to have an effect. Plants growing on top of cheese did better than the control, and liver did the worst. I hypothesized that this has something to do with the nutrients released upon decomposition. Cheese releases a lot more calcium, fatty acids, sodium, and phosphorus than liver does, whereas liver releases much more metals, like iron. Perhaps the cheese is serving as a fertilizer role, and the liver is actually introducing toxicity. This might also mean that cadaverine itself is not tied to plant death, as cheese is known to release higher concentrations of cadaverine into the soil than liver. Later this summer, I plan to do a field study using piglet cadavers as well as a depth test to attempt to answer the above questions. I also hope to continually photograph the whole cadaver experiment along with some liver, cheese, and control treatments using false color infrared photography, a kind of photography that can detect stressed vegetation. Stressed vegetation appears darker on the resulting photograph than non-stressed plants based on light absorbance, and so I want to quantify the degree of stress the plants are undergoing in a cadaveric environment using the reflectance-absorbance ratio, and see how cadaver-grown plants compare to liver, cheese, and a control. In addition, once I get lab access again, I would like to work with Dr. Hastings to get a good profile on what molecules cheese and liver respectively release upon breakdown. For germination, it was extremely obvious that the presence of decaying material resulted in no grow zone. This is in line with observations in the forensic literature that plants die in a zone around a decomposing body. The fact that a zone of inhibition was present during the germination test, but not the growth test, suggests that this cadaver island effect may be a germination inhibitor rather than a toxin, and this means that the observed dead zone may be the result of the body preventing the germination of weeds instead of killing off adult plants disturbed by the digging as previously thought. There is a strongly significant effect on soil pH caused by the decomposing matter, and it seems to be tied to what exactly is decomposing. Going forward, I'll run some germination tests involving piglet cadavers and varied depths to attempt to answer the above questions, and once I get lab access again, I'd also like to utilize my pure cadaverine from last semester and see if pure cadaverine itself prevents germination the same way, or if it seems to have no effect. Thank you for your attention. Support for this work is provided by the Haber Fund in support of the Haber Summer Research 2020 program. I also wish to thank Drs. Blome and Rivers, the Biology and Chemistry Department, and my family and fiancé for supporting my passion, as weird as it may be.